once again, good afternoon, everyone. So, before starting today's topic, let us revise some of the things which I have taught earlier. Like our topic was electrophoresis, so I'll go on brushing up the slides in order to make you recall the things which I have explained. And this was the outline of the talk, which on first class itself I have given. And if you recall, I told so Theselius, he was a scientist who first discovered the electrophoresis, and he also coined the term electrophoresis. He was working in the area of serum protein, and here, if you see the figure, so this is one of the Theselius apparatus. Then in introduction, I told you that this term has been coined from two different words. One is electro, that is electron or current, and another is phoresis, that is separation. That's why the electrophoresis, which is basically the movement of charged particles through any electrolyte when subjected to an electric field. And then some of the related terms, so that I introduced, like anaphoresis, cataphoresis, iontophoresis, zone electrophoresis, and even I introduced you to the basic principle of electrophoresis, that is electrophoretic mobility, which was defined as the rate of migration per unit field strength of the electric field, which we are going to provide. Then I also had given you the equation where we correlated different characteristic of the molecule with the electrophoretic mobility, like hydrodynamic size, shape of the molecule, pore size, viscosity of the buffer. Then I told you about the rate of migration and some of the variables which are directly or indirectly correlated with rate of migration. Then some of the factors which affect the electrophoresis, that also we discussed in length, like net charge on the particle, mass and shape of the particles. Then I told pH of the medium also affects the strength of the electric field, and then the supporting medium which we are going to use, and then the temperature. And then one by one I explain all these factors. Even I introduce you to the effect which is called as sieving effect, which is basically because of the pore size of the supporting medium which we are going to use. And I told how this is going to exclude or give a sieving effect to different shape of the molecule, which one will move faster, which one will move slower, and what will be the effect, overall effect on the movement, that we discussed. Then I told you about the buffer. So what is the importance of buffer and what type of buffer one should use. Then I had given you the list of some of the common buffers which we are using, and along with their pH, and why we are using some of the common that I explained you. Then we discussed about the supporting medium which we are using. And here, if you see this slide, I had given you some of the characteristic feature of supporting medium also, and some of the common supporting medium basically which people they are using in their lab. And then we selected two most common supporting medium, that is agarose gel and then polyacrylamide gel for the discussion. And in agarose gel, so I explained you that how this is, that agarose is basically a linear polymer of agrobios. And here you can see the structure also. And then I showed you the scanning electron microscopy of gel structure also. And right hand side, if you see, so how we used to cast and how this agarose is going to be polymerized. So all those informations was given. And then we discussed about the polyacrylamide gel, where I told you that this is also a monomer polyacrylamide gel. Hello. Hello. Hello.
Hello, sir.
Yeah. Shall I continue?
Yes, sir.
Yeah, so other than the agarose gel, I told polyacrylamide gel is also very popular, and most of the time this is basically preferred to be used for proteomic characterization. So here I also explained you about the structure, the polymerization of this particular polyacrylamide gel. What are the precautions one should take, how this is going to be polymerized, and what will be the pore size, and how the scanning electron microscopy structure of this polyacrylamide gel will look like. So all those informations were given. And then the most important slide where I told you about some of the important differences between the agarose and then polyacrylamide, which basically we are using as a supporting medium in most of the important molecules for separation. So different parameters were explained, and then pros and cons were discussed about these two, where I told that in case of polyacrylamide, you have a very smooth surface, whereas in case of acrylamide, the surface is not smooth. And then acrylamide, polyacrylamide is basically for protein, but then agarose is basically for DNA or RNA molecules. Then here I discussed you about the different types of electrophoretic unit which we are using. Like we have a horizontal unit, we have a vertical unit, and then different accessories which we are using along with that. Like you have a tank lid, casting dam, casting tray, gel tray, electrode, foam, etc. etc. Then so in second lecture, so I explained you the classification of electrophoresis, where I told you that the electrophoresis as such has been broadly classified into two different types based on the molecule which they are going to resolve. One is the technique or the unit basically dedicated for the separation of DNA, and other is the unit which is basically dedicated for the separation or the characterization of protein. So in case of DNA, I told you we have a very common technique, that is agarose gel electrophoresis. Then we have another technique which is called as sequencing gel electrophoresis, and I also told you that this is one of the preferred technique used for the sequencing. Then we have a pulse field gel electrophoresis, which is also called as a PFGE. And I told you that in case of high molecular weight DNA, so basically we used to prefer PFGE in order to get a high resolution where your DNA is going to be moved in a serpent way, and then after every 15 seconds or 30 seconds, we are going to give a pulse of electric field. And because of this, so the length of the DNA will be easily resolved, and then it will be separated onto the gel. Then I told you capillary gel electrophoresis a decade back. This was one of the popular technique which was preferred to be used for isoelectric focusing. So nowadays also we are using this, but because of the tedious process of casting and then running, so now very limited use of capillary gel electrophoresis is there. In case of protein-based technique, so I told you the most common technique is SDS-based, sodium dodecyl sulfate polyacrylamide gel electrophoresis. It uses another thing that I have explained you. Then I told that we have another technique which was basically discovered to have a in-gel activity assay, or to have a biological activity of the protein onto the gel. So that is called as NATipase. And then I told you we have four different classifications under NATipase. That is blue NATipase, blue native dyes. Here dyes stands for differential gel electrophoresis. Then we have a clear NATipase, which has been basically discovered for the metalloproteins. And then we have a QPNC-pase, which is basically utilized by the industry people. Then I told you that we have a technique which is very popularly used, that is gradient polyacrylamide gel electrophoresis. Here also, since I have not given, we have two different type of gradient. One is chemical based gradient, other is temperature based gradient. So maybe in subsequent slide or maybe next class, I'll explain you about the gradient polyacrylamide gel electrophoresis, where we are going to use a gel in a gradient. So purpose is different. And then we have a isoelectric focusing, which is basically based on the isoelectric point of the protein, which you are going to resolve. This is also one of the dimension of two-dimensional electrophoresis. So next technique, which is very popular, and this is also one of the most important techniques in gel-based proteomic characterization, that is two-dimensional electrophoresis. So here our first dimension, as I told earlier, is isoelectric focusing, where you are going to separate the protein based on the isoelectric point. And then the second dimension is the SDS phase, where you are going to separate your protein based on the molecular weight. Then we have another technique also, which is called as cellulose acetate electrophoresis. So in some of the method like dot blot and other lab-based methods, so there we prefer to use this cellulose acetate electrophoresis, but nowadays it is of not much use. And then we have a continuous flow electrophoresis, which is again basically preferred by the industry people for the separation of some of the antibody-based work. So they are using this continuous flow electrophoresis. Then I in length explained you about the SDS phase, and these are some of the uses which I listed you. Then we discussed about the basic principle of SDS phase, where I told you that there is an intrinsic charge on the protein, and then what is the purpose of using SDS, and how this SDS, which is basically a detergent, gives a unified charge to the protein, and how the reducing molecule which you are going to use, like beta mercaptoethanol or dithydritol, how it breaks down all the disulfide bond in order to linearize the protein, and then how your unified charge protein is separated on the gel based on the molecular weight. So that was explained here. Then I also explained you some of the characteristic feature of SDS, and even I use the note that in most of the cases, so the type of SDS which you are going to use is a very important one, because most of the time if there is a contamination in the SDS, and when you prepare your use this SDS for your SDS phase, most of the time you lose the resolution on the gel, and there will be a fuzzy, or there will be a smearing, or maybe proper band will not come on the gel. And then some of the characteristic feature of SDS that I explained here. Then I also explained you about the different instrument and accessories which we are using in case of SDS phase. And I told you we have three different types of unit which we are going to use. One is mini unit, which is a smaller one. Then we have a midi and large unit. And then I also told you what are the pros and cons of these units, and how much time these unit takes, and what is the cost of these units, and what will be the cost per sample for these units. So all those informations were given. Then I also discussed you about the protocol for the SDS phase, though this was not listed in the lecture, but then I thought that let us introduce this also, where I told you that we have four important steps when you plan to go for SDS phase. So here I explained you about the first step, that is protein extraction. So how you are going to extract the protein, what type of buffer one should use, and how you are going to quantify or quantitate the protein which you have isolated. So all those informations were given. Then gel casting, I told you that what are the different types of gel which we can cast, what type of instrument you are going to use, and then SDS phase run. So what are the conditions, what should be the current volt, all those information. Then staining, so I explained you about different types of staining, their detection range, and then some of the results which I show. This slide shows you about the extraction, which I explained you. I also told you about the buffer and different kits which are available for the extraction of a protein. And even I told you that the advantage of using kit is that it will not only help you in extracting the protein, but it will also help you in purifying the protein also simultaneously. Then I explained you about the different methods for the protein quantification, where I told you that we have a kit-based method also, like we have a Lorry's method, you have a Bradford dye, we have a non-interfering protein assay kit. So these are some of the kit-based method which basically people they are using. Most common out of this is Bradford dye, which we are using in our lab on a routine basis. Then I told you that now we have an instrument-based quantification of protein also. So the most cheapest one is nanodrop, but then the instrument cost is around 8 to 9 lakhs rupees. And this is basically emulsion-based method, where you are going to put a drop of the sample, and then it will tell you the quantity of the protein, or it is also used for the nucleic acid also. So it will tell you the quantity. Then we have a qubit, which I told you is proprietary of in vitro Zen, and here you have to purchase a standard and some of the dyes, and then easily once you feed your sample into a tube along with a buffer, it will tell you the quantity of the nucleic acid or the protein present in your sample. Then I told you that the advanced one is bioanalyzer. So here it will tell you the quantity also, and even the quality of the molecule which you have used for the quantitation. So these are some of the methods which basically people they are using to analyze or the quantitate the protein for quality analysis. So the best method is to run it on the gel, whether it is a protein or whether it is a nucleic acid. So this slide, I explained you about different type of gels. Even I use the term that this is also called as three gel system because we are using three different pH. Then polymerization also I explained you, and then I told you what are the pH. And in subsequent slide, so I raised the question that why there is a need to use three different pH, and then I explained you that what is the purpose of using three different pH, that is the pH of running buffer or stacking or resolving buffer. Then this was one standard table where I explained you that how a percent XL might affect the molecular weight range of the protein which you are going to separate, and every time based on the protein molecule which you are going to target, we have to decide on the percent acrylamide gel which we have to prepare. So then staining, so one by one few of the staining method which I explained you, like Comase dye stain. I told you this is one of the most common routinely used in the lab, and even I told you the detection limit which is basically 5 to 25 nanogram, and most of the gel SDS phase gel, so that is basically stained with the Comase dye. Then I also told you that at higher end we have a silver staining which is a little bit a costly method, and here you have a detection limit which is very like 0.25 to 0.5 nanogram, and time more or less the same, but then here resolution on the gel will be very high, and easily you can see that once the silver ion gets reduced to metallic silver, so easily you can see the brown or the black band color on the gel. Then I also explained you about the fluorescent dye staining. I told now this is picking up like anything, and now people they prefer to use this. Detection limit is more or less similar to the silver nitrate, and easy to use. Easily you can capture. Visibility is very good, and since we have a different type of fluorescent dye, so now people they are labeling these different protein samples with different dyes, and then they mix together and run it in order to get a beautiful mosaic of fluorescence on the gel. So a lot of combinations and permutations are there in case of the staining. Then I also explained you about some of the results, some of the problems when you run your sample on the gel. So some of the troubleshoot which you should know when you are planning to go for SDS phase, and some of the gel photographs I have shown here, like when there is a high current, what type of gel you are going to get, or when there is a partial run where there is a diffusion of the sample, or where gel is not properly casted, or where you have used enough of the protein sample than what is required, or where you have exceed or the transition phase of the stacking and resolving is not properly met. So in that case, what type of gel you are going to get? These are some of the limited information which I have given you. And then I told you that in next class, we are going to have a Nechi polyacrylamide gel electrophoresis. So this was in brief about the two classes which I have taken earlier. So let's start today with the Nechi polyacrylamide gel electrophoresis. This is also called as or denoted as N-phase. N stands for native. Native means we are trying to resolve the protein in its native structure itself. The purpose is that we want to analyze the biological activity of the protein per se when you are going to resolve it on the gel. So that is the purpose why the word native has been used. And polyacrylamide gel electrophoresis means the supporting medium which you are going to use here is a polyacrylamide gel. So if you see here, so the original N-phase system was first developed by Ornstein and Davis. So Ornstein and Davis in the year 1964. So he first discovered this native phase system out of requirement. So that time people they are doing a lot of this phase, but then Ornstein and Davis they were basically working on serum protein, and they wanted to preserve the native conformation and biological activity of the protein on the gel, and out of compulsion they discovered this particular method which nowadays we used to call it as native phase. So if you see here, I have given a notation that native phase which separates folded protein and protein-protein or protein ligand complexes by or based on the charge or the size or the shape. So maybe in subsequent slide, I'll tell you the principle and then other information. But from this slide, so you should know that this was first developed by Ornstein and Davis while working on the serum protein. And this was out of compulsion because they wanted to analyze the biological activity of this protein onto the gel, and they modified the procedure itself. And because of this, so they discovered this native phase system, and the term native has been used because we want to resolve the protein in its native structure. So this is a brief information about the native phase. If I use the term that SDS phase minus SDS is native phase, it will be not found. So in SDS phase, if you recall, we are using SDS in gel also, in buffer also, but in case of native phase, we are not using SDS. So SDS phase excluding SDS is basically native phase. So that's why in first sentence I've written native or non-denaturating gel electrophoresis. Non-denaturating means we are trying our best to create a condition where protein should not denature or aggregate. Because if it gets denatured or if it gets aggregated, so it will lose its biological activity or native conformation. So here our objective is to provide a continual condition where protein should regain its conformation and then biological activity. Now, second point is the mobility depends on both the protein charges and its hydrodynamic size. Protein charges means the intrinsic charge of the protein, which is basically because of the amino acid composition of the protein. So this is one of the basis for the mobility in case of native phase, and other is the hydrodynamic size, the size of the complex protein which you are going to separate. And here one more typical difference is that in case of native phase, in order to regain the native structure or the conformation of the protein, there is a compulsion that we have to run native phase at a temperature of 4 degrees Celsius. So basically we used to keep it inside the 4-degree freezer or maybe in a cold room to run this native phase. So this is one of the requirements when you plan to run a native phase. Third point says the electric charge driving the electrophoresis is governed by the intrinsic charge on the protein at the pH of the running buffer. So as I told earlier, so it is the intrinsic charge which is one of the basis in case of native phase other than the size of the protein which you are going to separate. And a fourth point is that intrinsic charge basically depends on the amino acid composition, which I have already told earlier. And so some of the post-translational modifications also modify the intrinsic charges of the protein, which is reflected in some of the movement or the resolution of this protein onto the gel in case of N phase. So second last point, if you see, higher mobility has been observed for more compact conformation and lower for large structure like oligomers, because sieving effect is also one of the prominent variables which decides the movement or the mobility of this protein in its native conformation. Some of the important uses, like one common use which basically in our lab or maybe in most of the lab people they are using is for zymogram study or for in-gel activity assay of different enzymes. So native phase is the preferred technique to use for in-gel activity assay of any enzyme or for the zymographic study of any enzyme, or it is also used to study the conformation of a specific enzyme or the self-association or aggregation of a particular enzyme or the binding of one protein with the other or the protein-protein interaction or the protein ligand binding. All those informations can be derived using different sub-techniques which has been modified and discovered under the native phase. So this is one obvious question because I explained you about the SBS phase and then now I'm explaining you about the N phase, that is native phase. So what are the basic differences between native phase and then SBS phase? So some of the differences which in between I have already explained you that one common difference is that in case of native phase we are not using the SDS, whereas in case of SBS phase, as the name suggests, SDS is one of the prominent detergent which we are using. In case of native phase, also we are using detergent, but those detergents are basically non-ionic detergent. Whereas in case of STS phase, you are using an ionic detergent for the separation. Second is in case of native phase, as you know, the basic principle that separation is based upon the charge or the intrinsic charge of the molecule and size and shape of the molecule, whereas in case of STS phase, it is based on the molecular weight of the protein. Then in case of native phase, so some of the sub techniques of native phase are there where easily you can separate or purify the mixture of protein by running it onto the native phase cell in its biological conformation. Whereas in case of SDS phase, the most common utility of this technique is to find out the molecular weight of the unknown protein which you are going to use. And native phase, I think this was the original method which was discovered for the electrophoresis. Whereas SDS phase, this was basically discovered to check the purity of the protein which you have isolated. So these are some of the differences. Other than this, few more differences are like in case of native phase, I told, so neutral or non-ionic detergent is used, whereas in case of SDS phase, you have a charge or anionic detergent is used. In case of native phase, there is a requirement that the run should be done at 4 degrees Celsius, whereas in case of SDS phase, you can run it at room temperature also. In case of native phase, so basically this is also called as a single gel system, because in case of native phase, we are using a resolving gel only, and then most of the time stacking gel is not preferred in case of native phase. Whereas in case of STS phase, as you recall, I told this is also called as a 2-gel system where you are going to use a stacking gel as well as a resolving gel of different hues. So these are some of the important differences between native phase and STS phase, and you should know. So coming on to the basic principle of this native phase, as I already explained you about the electrophoresis basic principle, then STS phase basic principle. So it's not difficult to presume what is the basic principle behind the native phase. So here proteins are separated according to net charge, size, and shape of their native structure. So electrophoretic migrations occur because most of the protein, they carry a net negative charge in alkaline running buffers. Third point says the higher the negative charge density, the faster the protein will migrate. And last point is frictional force of the gel matrix, the pore sizes or the sieving effect which happens because of the gel matrix. So it also regulates the movement of the protein according to their size and three-dimensional shape. So these 3 points are basically the deciding or the basic principle behind the movement of the molecule in case of native polyacrylamide gel electrophoresis. Now coming on to the classifications of native phase. So native phase has been broadly classified into 5 different types. First one is N phase, where I have written this is basically normal native phase, the basic native which we are using in our lab. So that is N phase. Then you have another method which is very popular and a lot of publications are now coming and people they are using this specifically for membrane bound proteins. So this is called as blue native phase. In short notation, we also used to call it as BN phase. So blue native polyacrylamide gel electrophoresis. Then we have a third method which is also called as blue native dyes. So in short notation, we used to call it as BN dyes. Here dyes stands for differential gel electrophoresis, and this is modification over the BN phase. Then we have another method which is called as clear native phase, CN phase. And as I told earlier also that CN phase is a preferred technique to be used for the metalloprotein. So the only difference is that here the polymerization time is very long, like you have to polymerize, you have to keep the gel for polymerization maybe up to 62 to 69 hours. And once it polymerizes, then only we can use it for the separation of metalloprotein in its biological conformation. And last method is QPNC phase. So as I told earlier, this is also one of the methods which is very preferred and preferred to be used by the industry people. So let's take one by one these methods. We have one more classification which is basically based on the supporting medium which you are going to use. So native base has been broadly classified into two different types based on the supporting medium which you are going to use, that is polyacrylamide and agarose and differences. So polyacrylamide, so this is basically preferred to be used where you are going to separate a protein of 5 to 2000 kilodaltons, whereas agarose is preferred to be used in case where you have to separate a protein of more than 200 kilodaltons. In case of polyacrylamide, as I told earlier, you have a uniform pore size, whereas in case of agarose, so pore size is not uniform. And one more important information is that polyacrylamide, as you all know, is a potent neurotoxin, so both of these are potent neurotoxin, and we should take utmost care while you are using this. So polyacrylamide, starting from the powder to the polymerization to the throwing, you have to take utmost care. So native base has been broadly classified into 5 different types, as I told earlier, based on its utility, and into 2 broad types based on the supporting medium which you are going to use. So let's have a look on the first, that is normal native base, which is basically used in most of the labs. So this normal native base, as I told, so this is SDS base without SDS and then reducing agent. Here separation is based on charge to mass ratio. Here we are not using any denaturating or strong denaturating agent, and here separation is based on the size, shape, and native charge of the molecule. So here, if you see in box, I have shown intrinsic charges and then hydrodynamic sizes, which is the basic principle of this N phase. And in case of intrinsic charges, I have written that amino acid composition and then post-translational modifications besides the intrinsic charge of the protein, which you are going to separate in its biologically active form. This N phase is very popularly, yes, so this shows some of the uses of this N phase. This is very popularly used to characterize the isoenzymes of or the activity of isoenzymes on the gel, or different types of isoenzyme of a particular enzyme. So all those characteristics you can do easily on the gel. This normal native phase is also preferred to study the conformation of the protein. So what type of conformation a protein has, that easily you can track by running it on native phase gel. It is also used to study the protein-protein interaction. And this technique is also used for self-association or aggregation characterization of any protein. So easily you can run it on the native phase gel and you can analyze whether the protein can self-aggregate or what type of aggregation of protein takes place. So that easily you can derive by running it on the native phase. This method is also used to recover protein from the gel, because here we are not using any specific detergent, so easily you can recover that protein in its biological form from the gel. You can dilute it in some buffer, and then in vitro you can analyze the activity of that specific protein in order to find out whether the recovery was perfect or not. So this method is also preferred to use to study the protein ligand interaction. So this method has a lot of literatures are there where people, they have used this native phase to characterize protein ligand interaction. So we are also working on this, and basically we are using this to study in-gel activity assay. So maybe next class I'll show you some of the results which we have derived here in wheat crop. So coming on to next method, that is a blue native base, as I told. So this blue native base, as the name suggests, blue native. So this blue basically has been coined because of the use of Comase blue dye. Comase blue G250 dye we are using here. So we are going to label the protein using the Comase blue G250 dye. Here, if you see this BN phase or blue native polyacrylamide gel electrophoresis, it resolves the protein complexes by molecular weight while retaining their native structure through gel electrophoresis. So it will resolve the protein based on its molecular weight while resolving it on the gel. The only thing is, in place of SDS, or is for the substitute for SDS here is say blue G250 dye, which we are using to coat the protein, which provides the necessary negative charge for the migration to the anode. So here, if you see, so we are going to use an PBB dye and along with the protein. So protein is labeled, and then you are going to run it like what we used to do in case of SDS paste. This BN paste nowadays, if you search on net, so a lot of publications are coming out in journal of higher repute like Nature and other, where people they have used this BN paste to characterize some of the organelle proteins or some of the membrane proteins present in different organelles. So there they have used this technique to characterize this complex supraprotein. If you see, this particular technique was discovered by Shager and Jago. So Shager and Jago, they have their own publication also. So these two people, they discovered this modified form of native paste, that is blue native paste. Again, let me repeat that this blue term has been coined because we are using CBB dye, that is common blue dye, and native paste because we are running it maintaining the conformation or the biological activity of the complex protein molecule which you are going to separate. Here you can separate a protein between 100 kilodalton to 10 megadalton. So even a big supra-complex protein also you can easily resolve using BNPs. And here if you see that the most important use of this BNPs is for protein-protein interaction. So easily you can track and characterize the protein-protein interaction using the BNPs. This is also used for the separation of enzymatically active protein complexes under mild conditions. So here the condition is not stringent as in case of SDS. In order to maintain the conformation of the protein, and easily you can load a supramolecule protein and you can analyze the enzymatically active proteins onto the gel. This is also used for the analysis of hydrophobic membrane proteins. So for hydrophobic membrane proteins also, this is one of the preferred techniques. So these hydrophobic proteins, they have a specific characteristic that it can easily solubilize the non-ionic detergent, especially tritone X100, digitonin, etc. So out of these two, digitonin is preferred over others. So we are using in BNPs, we are using non-ionic detergent, we are using a CBB dye to label the protein, and then we can resolve the protein like what we are doing in case of SDS phase. The only thing is here CBB is used as a substitute for the SDS, and this method is basically used to characterize the membrane protein or hydrophobic protein or the enzymatically active protein complexes onto the gel. So coming on to the another method, which is basically a modifications over a BN paste, that is blue native paste, that is called as blue native dyes. Here dyes stands for differential gel electrophoresis, and here we prefer to use a cyanine dye, which is basically a water-soluble fluorescent dye. So there are different formulations of cyanine which are available, and on excitation it gives different colors. Like it excites at 550 nm and gives a green color, and then we have different type of cyanine also denoted as Cy 2, 3, 5, 8, 10 like this, and each has a different excitation colors. And since these are water-soluble fluorescent dyes, so easily you can use this for labeling the protein. So protein, in case of BN dyes, we are going to label different protein with different cyanine dye, and then you are going to run it on the gel. And left-hand side, you can see the beautiful mosaic of different protein spots which are visible onto the gel. So easily you can differentiate different samples, their location, their run, their resolution, everything you can locate onto the gel. And the purpose of discovering this BN dyes is that it eliminates gel to gel variation, because when you are running one set of gel for one sample, second set for second sample, so most of the time the condition will vary. The run condition, the gel condition, and other variables will vary. But when you run it on the BN dyes, by labeling all those samples using different signing dye and then running it in one gel. So easily you can eliminate the variations and then easily you can locate different samples onto the one gel. So this is the beauty of blue native differential gel electrophoresis. Uses same as I explained in case of blue native paste. Basically it is preferred for membrane protein or supramolecules or for trapping the biological activity of this membrane protein onto the gel. Coming on to the another method, that is a clear native paste. So this CN paste is also very popular, and as you know, this was discovered by Milder. So he first discovered this CN paste, and basically he wanted to analyze the enzymatic activity or the catalytic activity of the enzyme onto the gel. So to achieve this objective, he discovered this clear native paste. So here you can separate acidic, water-soluble, and membrane protein with isoelectric point less than 7. So all those proteins like acidic or water-soluble or membrane protein, you can easily separate using the clear native paste. It has a lower resolution than blue native paste. So if you compare CN paste with blue native paste, so in case of blue native paste, the resolution is high as compared to clear native paste. And as the term suggests, clear native means here we are not using combustible brilliant blue dye as in case of BN paste. And this CN paste was basically discovered to determine the catalytic activity of the protein molecule onto the gel. So using BN paste, because of combustible blue, which gives the anionic charge to the protein, so it's very difficult sometimes to analyze the catalytic activity of the protein. So in order to in order to remove the interference of this combustible blue, which is one of the prominent member in case of BN paste, so this CN paste was discovered, clear native paste was discovered where we are not using combustible blue, and then easily we can remove the interference which is caused due to combustible blue, and then easily you can use it to determine the catalytic activity of the enzyme. Even in case of FRET analysis, FRET is a Foster resonance energy transfer. This is basically to characterize the structure of the protein. So in case of FRET analysis also, prefer to go for CN paste rather than blue native paste, because in case of FRET analysis also combustible blue interferes. This is also used to analyze the enzymatically active state of enzyme. So this CN paste, you can easily analyze the active state of the enzyme. You can separate it, you can detect it on the gel. So all sort of things you can do on the gel. CN paste is also a modification over the blue native paste. So CN paste is basically a blue native paste without combustible. So coming on to the last technique of native paste, that is QPNC paste. So this QPNC basically stands for a quantitative preparative native continuous paste. Quantitative preparative native continuous polyacrylamide gel electrophoresis. So as I told earlier, it takes almost around 69 hours to polymerize the gel. And here you have a minimum molecular saving action. So because of this long duration and low concentration of the gel, polyacrylamide gel, which you are going to cast, it has a minimum molecular saving effect. Pore size is very large, and it is basically used to separate acidic or alkaline or neutral metalloproteins. And here the molecular weight which you can separate is 6 to 200 kilodaltons. So this is the molecular weight which you can use to separate, and then retention of the structure and function of metalloproteins can be easily retained onto the gel. And hemoglobin, which is also a metalloprotein, was first characterized using QPNC paste method only. Hemoglobin, so which is basically a metalloprotein. Another metalloprotein also people they are using this in industry. So this is basically used by the industry people. Left hand side you can see the apparatus which is very complex. So this is the apparatus of QPNC paste. In a lab, so very few labs they have this facility. Most of the time industry people they are using this for their commercial purpose. So this was in brief about the native paste as well as the other methods which I explained. If you have any queries, please ask. If you have any question, any queries, please ask. Hello.
Hello, sir.
Yeah.
So it's called blue native differential gel electrophoresis because we're using different dye. Because of that reason, it's called differential gel.
No, no, not different dyes. So BN dyes is basically dye stands for differential gel electrophoresis, and here you are using a water-soluble cyanine dye, right? So other than the Comase Brilliant Blue, you can use a cyanine dye which gives different colors. Now suppose if you have a 4 samples of protein which you want to resolve it on the gel, right? So suppose sample 1 you have dye with, you have used the dye cyanine 1, in sample 2 cyanine 3, in sample 3 cyanine 5, like this. So when you mix all these samples and run it on the gel, so when it resolves, so you will get different fluorescent color coming out of those samples. So this is the purpose of BN dyes, and why we are using blue native, because all other processes or steps we use to follow is of blue native phase only. The only thing is here, the extra is that we are labeling it with water soluble dye in order to get a different colors.
Okay, sir.
Okay. Hello.
Thank you, sir.
Oh. Any other queries? Any other questions? Even you can write your questions on this theme also. So there is a chat, this one, where you can write your questions, and then I will love to answer.
Sir,
Yes.
is this FRET analysis can be used for separation of nucleoproteins?
Which one?
FRET analysis using CN page.
Clear native base is
Yeah.
used for.
Sir, this FRET analysis is used for separation of the nucleoproteins, no?
Your voice is not. Can you repeat?
Sir, FRET analysis.
Yeah. FRET is basically a this is a technique. This is an instrument. FRET it basically stands for Foster Resonance Energy Transfer, right? So basically used to analyze the structure of the protein.
Sir, this is used for separating the nucleoproteins also, no?
No, FRET you cannot use for the separation of anything. FRET will just give you a value which you can correlate with the structure of the protein which you have used for the FRET analysis. For separation, you have to use any gel-based method only, and native phase is one of the methods where easily you can separate the protein in its native or biologically active form.